Hello, I am Kunchumohan Vadakan. I study the nervous system to understand how it operates. I have a conflict of interest to declare. In this presentation, we will examine a potential mechanism for the action of anesthetic agents. Anesthetic agents were discovered more than 150 years ago, but still we don't know how they act. We have very large number of information that we have accumulated from studies using anesthetic agents. If we look at linear algebra, we can see that they are solving a system of equations having more than one unknown variable. If there are more equations than the number of unknown variables, then we can solve a system of equations. Here, in the case of anesthetic research, we have very large number of findings and we have many unknown variables. So, taking inspiration from linear algebra, we can try to attempt to solve the system. In the case of anesthetics, they reverse the level of consciousness, but we don't know the normal mechanism of consciousness. So, if we have a framework for consciousness, then we can examine whether all the findings in anesthetic research are they fitting with the proposed mechanism of normal consciousness. So, we can do this exercise and figure out how the anesthetics work. So, first let us enumerate all the key findings that we can use as pieces of the puzzle to solve the system. First, anesthetic agents act on large number of membrane bound receptors. Some of the anesthetic agents have agonist action on different receptors such as GABA receptors, opioid receptors, alpha adrenergic receptors, and some of the anesthetics have antagonistic effect on receptors such as NMDA receptors and dopamine receptors. So, if we look carefully, they are all different in nature, yet they all produce loss of consciousness. But the only common thing among them is the fact that they are all membrane bound receptors. That is one piece of the puzzle. Then we have more pieces here. That is, anesthetic agents are lipophilic in nature. They are all lipophilic in nature. In fact, inhalational anesthetics were studied for their potency versus their lipophilicity. If there is a graph of their potency versus partition coefficient in olive oil versus water, this part shows the lipophilic nature. This correlation between the potency and lipophilicity show a very robust correlation coefficient, that is 0.997, and that correlation coefficient is the highest in biological systems. So, this shows that as the lipophilicity of the anesthetic agent is high, those anesthetics show very high potency. This is in the case of inhalational anesthetics. And the third finding is that if we anesthetize an animal and put that animal in a box, and if we increase the pressure within this box, we can use either aquatic animals or terrestrial animals. So, we can increase the hydrostatic pressure or the gas phase pressure. In either case, the anesthetized animal's loss of consciousness reverses very fast if we increase the pressure. So, there is reversal of anesthesia when we increase the pressure, and we don't know why that happens. This is a piece of the puzzle. Now, another finding is if a person is exposed to multiple general anesthesia, then there is a likelihood that that person can suffer neurodegenerative disease. So, why neurodegeneration? That can be taken as a good piece of the puzzle to solve the system. Finally, we can see that normal level of consciousness is related with the frequency of the oscillating extracellular potentials. So, there is a normal frequency, and that frequency is required for maintaining normal level of consciousness. When we use anesthetic agents, this frequency goes down. Frequency means the number of oscillations per second. So, how these anesthetic agents cause reduction in the frequency of oscillating extracellular potentials and how that can lead to loss of consciousness. Now, if you look here, we have enough pieces of the puzzle here, and if we can find a mechanism for normal consciousness, and if we can explain all these features, then there is a good chance that that hypothesized mechanism is correct. So that's the whole exercise that we are going to do here. I have explained a framework for consciousness in a separate presentation. Here I will briefly explain it once again, and then we'll examine all these key pieces of the puzzle. All the higher brain functions such as consciousness, perception, memory and thought processes, they are all first person inner sensations. Let us examine why the nervous system is generating first person inner sensations. It's a virtual sensation. So here we have to find out whether there is anything that is present within the nervous system that has the ability to associate various sensory features when the item is close to that nervous system, and whether that associative feature can be reactivated by the arrival of one of the sensory stimulus to induce inner sensations. That's the exercise that we have to do and find out. So that's what I'm going to do here. So let's figure that out. So here item is close to the nervous system, and here we can see the item far away from the nervous system. Let us draw it here and find out. So here is the sensory stimulus S1 that is coming to this presynaptic terminal, and here is the postsynaptic terminal. In other words, it's also called spine or dendritic spine, and this junction is called the synapse. When the activity arrives here, it will depolarize this spine head, and that depolarization will propagate towards its neuron, and it can propagate through different neuronal orders, and finally it may end in an end organ. Sometimes it can be a motor unit. If this neuron is remaining at a subthreshold level, then the arrival of this depolarization can add on to that subthreshold depolarized level and can trigger an action potential, which will allow that activity to propagate downstream. And we can also imagine that these neurons are also at subthreshold levels. There are a lot of regulatory mechanisms which I am not going to talk today. Similarly, we have the second stimulus S2 that comes to its presynaptic terminal, then to its postsynaptic terminal, then propagates towards its neuron N2, and it can reach its end organs. If these two sensory stimuli converge at this point, and if at the location of convergence, if the associative learning can induce a change that can be reactivated when the item is far away from the animal, that's what we are looking for. So based on semblance hypothesis, we expect a change to occur between these two dendritic spines, and those changes can be of different types. Number one is exclusion of water of hydration. Normally these dendritic spines are separated by water of hydration, so there are no electrical connections between them. Once there is exclusion of water of hydration, an electrical connection forms between them so that depolarization can spread between these dendritic spines, and further association can lead to formation of a membrane hemifusion, that is both partial and complete. So we can draw a picture like this. If you take these postsynaptic membranes alone here, we can get a picture like this after membrane hemifusion. Instead of 4 layers of membrane here, we have only 2 layers. That means these 2 membranes have undergone a Hemi fusion here. These two types of changes are expected to occur during associative learning event. Now, let us see whether this change can be reactivated when the item is away from the animal and whether that can induce inner sensations. So, here the item is away from the animal, let us draw it here. Now, the question is, when the item is away from the animal and when one of the sensory stimuli S1 arrives at the nervous system, can this S1 induce inner sensation of S2 is the question. Is there anything that is present at this location that can give rise to the units of inner sensations of S2? Based on Semblance hypothesis, an interpostsynaptic function link is expected to form between the dendritic spines and not anywhere else. This can be explained as follows. On a background state, there is quantal release of neurotransmitter molecules from this presynaptic terminal towards the synaptic cleft, which will depolarize this dendritic spine head. So, a portion of this dendritic spine head is going to get depolarized all the time, 24 hours, even while we sleep. And if we plot it here, it can be represented like this. There is small depolarization of the spine head taking place all the time. And occasionally, an action potential induced depolarization will propagate towards this presynaptic terminal, and that will cause a release of a volley of neurotransmitters, and that will lead to postsynaptic potential, which is a large potential. So, that is the background state, and that is occurring at the synapse as well. Now, during the background state, there is quantally released neurotransmitter molecule induced depolarization of the spine head all the time. So, that is the background state. In that background state, the S1 is arriving. It depolarized the spine head, depolarization spread across this interpostsynaptic function link, and it will depolarize the spine head. Now, what is going to happen at that point is that this depolarization that is coming incidentally will trick this dendritic spine to hallucinate that something is coming from its presynaptic terminal and from the sensory receptors of S2. So, that is what is expected to happen, and that is a cellular hallucination. So, you might wonder why cellular hallucination, because until here and until here everything was real, but now all our higher brain functions such as perception, consciousness, memory, thought processes, they are all first person virtual inner sensations to which only the owner of the nervous system has access. So, we have to go and find out a mechanism that can generate virtual inner sensations. That is the difficulty that we had in the past, and here we have a candidate mechanism. And the reason for induction of inner sensation has to be discovered. So, here is the potential mechanism, and I will repeat it once again. There is quantal release of neurotransmitter molecules occurring all the time from presynaptic terminal and depolarizing the spine head 24 hours all the time. Occasionally an action potential will arrive here that will lead to a huge potential that will propagate down towards the neuronal soma. So, that is the background state. In that background state, now the item is away from the nervous system, and one of the sensory stimuli is arriving here, depolarizing the spine head. Depolarization is spreading through this interpostsynaptic function link and arrives here. So, in this background state of depolarization occurring from the presynaptic terminal here, there is an incidental lateral activation which will trick this interlinked spine to hallucinate that it is receiving sensory stimuli from its sensory receptors. So, that is the cellular hallucination part. In other words, there is a semblance of arrival of activity from this presynaptic terminal and in turn from its receptors. So, therefore, this hypothesis was named as Semblance Hypothesis. So this is proposed to occur as a systems property of systems that have oscillating extracellular potentials. That means when we put two electrodes into the extracellular matrix space, and if we measure the difference in the potentials between the tips of those electrodes, and if we plot that difference, that's going to oscillate, and the frequency of that oscillation is very important. Only at a specific frequency that the system will induce cellular hallucinations. Now we have to go and find the content of that cellular hallucination in order to understand whether this S1 can induce the hallucination about S2. Can this hallucinate has the specific characteristic features of S2 is the question. For that, we have to make a retrograde extrapolation from this interlinked spine towards its sensory receptors. So the question is, activation of what sets of sensory receptors will activate this postsynaptic terminal in normal situations. So we have to do that extrapolation back from here towards the sensory receptors. So everything from here upwards is virtual in nature, and we had no experience in dealing with this situation in biological sciences in the past, and therefore this is going to be a difficult situation in the beginning. But as we continue to proceed and continue to test this hypothesis, we are likely to become comfortable in this area. We have to explore and find out the content of that cellular hallucination. So everything from here onwards is virtual in nature. We cannot verify this using biological systems. We have to make a hypothesis, and we have to build a framework for how this is occurring, and then we have to replicate this mechanism in an engineered system for verification. Once we do that, we know what's going on in the biological system. So that's a challenge in front of us. This postsynaptic terminal is receiving activity from its neuron, and we know that this neuron can fire an action potential when nearly 40 to 100 of its dendritic spines are activated out of nearly 10 to 30,000 inputs that it already have. In our extrapolation, we will see a large number of dendritic spines of this neuron, when activated, can give rise to activation of this postsynaptic terminal. So if we continue to extrapolate back towards the lower orders of neurons, then we will see much larger numbers of neurons, and finally we will reach the sensory receptor level. That's a large sensory receptor area. Now we have to ask the question, do we really need to activate all those sensory receptors in order to activate this postsynaptic terminal? We need not necessarily have to. Only a subset of the sensory receptor activation should be able to activate this postsynaptic terminal. We need a packet of sensory stimulus to activate that set of sensory receptors, and let us call that virtual packet of sensory stimuli as semblance. So if we know the structure of the nervous system, we can extrapolate back and find out the semblance, and there are many semblances as there are many sensory receptor subsets that can activate this postsynaptic terminal. Using this knowledge, we can try to replicate that mechanism in an engineered system. When the sensory stimulus S1 arrives, it's not only going to induce semblance from this interlinked spine, it's going to generate it at many such interpostsynaptic functional links. So for example, if we can put this and represent this as a triangle with many semblances here, this is going to happen at many levels, and so we have to represent them graphically like this. It's going to happen at many neuronal orders. We don't know the nature of that computational process, but one thing is for sure, there is an overlapped region here, and we can go and examine whether this overlapped region is giving rise to the sensory characteristic features of S2, and that is what we are expecting, that the system is going to hallucinate. The net product of all semblyons to give rise to S2. We have to figure out the mechanism of that natural computational process that is occurring at physiological time scales. Now we have defined a mechanism for generation of inner sensations. Now we have to ask the question, what is the inner sensation of consciousness? From this generation of inner sensations, can we predict how that may be occurring? If this nervous system is surviving in an environment, this nervous system is going to associatively learn large number of inputs from the environment. Many inputs that are arriving from the environment are coming at a background state. For example, gravity is there everywhere. I am not feeling the weight of my legs. My nervous system cannot afford to generate inner sensations towards all those sensory stimuli that are arriving towards me. If that is the case, my nervous system is not going to survive in this environment. I have to exclude all the background sensory stimuli that are arriving towards my nervous system so that the nervous system can pay attention to those specific deleterious or beneficial sensory stimuli that will allow my survival in the environment. My nervous system is not going to focus on generating inner sensations to the common sensory stimuli that are arriving towards my nervous system. So if a nervous system can do that, that nervous system can continue to survive in the environment. So the question is how to suppress the generation of inner sensations to all the common sensory stimuli. That can be done as follows. Even in the absence of arrival of those stimuli, if those interpostsynaptic functional links can be reactivated continuously, and if all those can generate a net semblance. So in that net semblance, the nervous system is not going to generate independent, individual inner sensations to the common sensory stimuli. So the question is how the system can generate a common net inner sensations to all those common sensory stimuli from the environment in which the nervous system is surviving. For that, we have to look into the nervous system, what happens to it when this nervous system continuously learns. So here we have these two postsynaptic terminals. Let us make a cross section here and put these two dendritic spines here. There is an interpostsynaptic functional link between them. During continuous associative learning, this is going to generate large number of interpostsynaptic functional links. There are very large number of associative learning events. They are all separate dendritic spines that are interlinked to form an islet of interlinked dendritic spines. Our idea is to generate a net semblance from all the common sensory stimuli, even without the arrival of those stimuli from the environment, because the nervous system wants to avoid inducing those regular inner sensations. For that, these interlinked spines that receive common sensory stimuli from the environment has to be activated all the time. So do we have anything for that in a background state? That's the question. If you look here, if this is the cortex and these are cortical neuronal orders. So here we have cortical neuronal orders 1 to 6, and from the 6th order outputs goes to the thalamus. So this is corticothalamic pathway, and from the thalamus inputs arrive towards the layer 2 and 3, and there is a reverberatory circuitry that's going on between the cortex and the thalamus. Between the thalamus and the reticular activating system, there is a reverberatory circuitry that's going on all the time, and the thalamus is receiving sensory stimuli from most of the common sensations. So now we have a reverberatory circuitry where the thalamus is receiving common sensory stimuli and everything else is reverberating. And in this particular instance, if these interlinked spines that receive common sensory stimuli from the environment is getting reactivated to induce background net semblance. Say for example, if these are the interlinked spines, and if that is the semblance that are arriving from one group of interlinked spines, and if this is from another group, and if we can put all the islets of interlinked spines, and as a result of this reverberatory circuitry, if they can all get activated to induce a common net semblance, then we can call it as C semblance for the generation of the inner sensations of consciousness. In that background state, the system will not generate separate inner sensations to separate background sensory stimuli. So between these cortical orders, we expect these groups of interlinked spines that are formed as a result of background sensory stimuli. And if they can be reactivated, the system can generate a common net semblance from all these islets of interlinked spines that can be thought as the net C semblance responsible for the generation of inner sensation of consciousness. So now in this background state, if the animal is receiving a unique stimuli or a beneficial or a deleterious stimulus, then it can induce specific inner sensations for that, so that the nervous system will become much efficient in the environment. That is the hypothesis of the generation of the inner sensation of consciousness. Now we have to go and examine whether this hypothesized mechanism of generation of the inner sensation of consciousness can explain all the pieces of the puzzle here. Even though different anesthetic agents act on different receptors that are having different mechanisms, and still they all cause a common feature of loss of consciousness. Now in a second I will explain how these anesthetic agents can reverse the net C semblance responsible for inner sensation of consciousness. Potency of anesthetic agents depends on the lipophilic nature. That means more anesthetic agents bind to the membranes, their actions will be more. It is already known that anesthetic agents change the curvature of the membranes. Now if we look at this interpostsynaptic functional link here, we know there are two mechanisms. One is exclusion of water hydration and membrane hemifusion. If the anesthetic molecules bind to this membrane and change the curvature of the membranes, it can facilitate the formation of membrane hemifusion. So we can expect large number of non-specific interpostsynaptic functional links, non-specific interpostsynaptic functional links to get formed during anesthetic action. So if large number of non-specific interpostsynaptic functional links are formed, what it does is that it will change the frequency of oscillating potentials. If we see an oscillating function, we can decompose that oscillation into its vector components. One vector component and another is a perpendicular component. Now we can explain the vertical component as contributed by the synaptic transmission in this direction, and the perpendicular component is contributed by the transmission of potentials across the interpostsynaptic functional link. These two components contribute to the oscillating extracellular potentials. Of course, there are additional contributions. One is this reverberatory circuitry, then we have recurrent collaterals that also contribute to these oscillating potentials. Now, if the anesthetic agents are binding to these membranes and generating large number of non-specific interpostsynaptic functional links, that's going to increase the horizontal component. That means this component is going to get increased, and if that is the case, then the oscillating frequency will reduce to lower levels. Now that is what exactly we are finding during anesthesia. The frequency of oscillating extracellular potentials reduces. Now what will happen for the C semblance? As the oscillating extracellular potentials reduce, the conformation of C semblance collapses. It completely changes. Once the conformation of C semblance is altered, we lose consciousness. We lose that background state of inner sensations upon which we can make unique inner sensations. That can explain why there is loss of inner sensation of consciousness in the presence of anesthetic agents. Now we have to explain neurodegenerative changes. Anesthetic agents are expected to form large number of non-specific interpostsynaptic function links. Now, the normal mechanisms of interpostsynaptic function links are exclusion of water or hydration and the formation of membrane hemifusion. What we know is that membrane hemifusion can further advance forward to form membrane fusion. So now this hemifusion has progressed towards a fused state. Now based on Semblance hypothesis, the interpostsynaptic function links are occurring between the dendritic spines that belong to different neurons, because on a dendritic branch there are dendritic spines, and it is known that the diameter of these dendritic spines is on an average the inter-spine distance. So if that is the case, then the interpostsynaptic function links should occur between dendritic spines that belong to different neurons. So now, because of the presence of anesthetic agents, if the interpostsynaptic function link here, which is a hemifusion, if that progress to a fused state, the cytoplasm between these two dendritic spines are going to get mixed, and these two are belonging to different neurons. This is neuron N1 and this is neuron N2. And even if these neurons are neighboring neurons within a neuronal order, it is clearly known that the mRNA expression profiles, even among neurons that are neighboring within a given neuronal order, are different. So since the mRNA profile is different, the protein expression patterns are also expected to be different. So if cytoplasms between these two neurons are mixed, then there will be cellular response reactions towards that. One will be precipitation of protein. Proteins can precipitate, and there will be a natural attempt from the cell side to close this fused area. If that fails, then the cells might initiate certain mechanisms to detach this spine, and we may see loss of dendritic spines. So loss of dendritic spines is a characteristic feature seen in neurodegenerative diseases, and protein precipitation is also another characteristic feature. If that also fails, then this neuron will undergo an apoptotic or a degenerative state, so that at least by sacrificing this neuron can survive. So such cellular mechanisms can initiate, and these are seen in neurodegenerative disorders. So we can explain that feature. And then finally, the pressure reversal. How can we explain that increasing either the hydrostatic pressure or the gas phase pressure of an anesthetized animal put inside a box will cause quick reversal of anesthesia? How can we explain that? If we can explain that based on the mechanism of generation of inner sensations and generation of C semblance, then we have a candidate mechanism here. So how can we explain the pressure reversal of anesthesia? It can be explained as follows. Here is the animal that is anesthetized and it is kept in a box. And if we are increasing either the hydrostatic or the gas phase pressure. So it can be an aquatic animal or a terrestrial animal which is anesthetized. We are increasing the pressure. Now we know that the anesthetic molecules are binding to the lipid membranes and it is causing large number of non-specific interpostsynaptic functional links that is altering the net C semblance and causing loss of consciousness. We have to make these anesthetic molecules to escape from this postsynaptic terminal in order to bring back those postsynaptic membranes to the normal state. How to get those anesthetic molecules out of the membranes is the question. This explanation became possible only by the recent discovery of paravascular space within the nervous system, which is called glymphatic pathway. The increased pressure within this chamber is acting through the tympanic membrane, and that pressure is getting transduced through the perilymph, through the cochlear aqueduct and endolymphatic duct towards the CSF. CSF runs through a paravascular space around the penetrating arteries. So through that paravascular space, that pressure can get transduced into the extracellular matrix space. This is the extracellular matrix space, the space around these dendritic spines. When there is excess pressure here, and if that pressure can further transduce outside, then these molecules can escape through that pressure difference. Now, from the extracellular matrix space, pressure is getting transduced to the paravenular space, and from there to the veins and to the lungs, and anesthetic molecules can escape into the chamber. In summary, when we put an anesthetized animal into this chamber and increase the pressure within the chamber, the pressure is getting transduced towards the CSF and to the extracellular matrix space, and we are letting the anesthetic molecules to escape from these membranes and to get equilibrated within the body of that animal, and again equilibrate with this space within the box. So that way we are trying to remove anesthetic molecules from the membranes as fast as we can. So that can explain how, by increasing the pressure within a closed chamber, can reverse anesthesia in an anesthetized animal. Now, using the proposed mechanism of generation of inner sensations and the generation of inner sensation of consciousness, we have explained all these features. And before this explanation, all these features were unrelated. And now this mechanism allows us to interconnect and explain all the diverse set of features that we had difficulties in finding explanation. So I sincerely hope that we have a testable mechanism of nervous system function here that can explain the action of anesthetic agents. I hope that you understood these explanations. If you have any questions, please feel free to ask me below this presentation or email me. I will be happy to answer them as much as I can. Thank you and have a great day.